Alright guys, welcome back. We're still on chapter 54. We're going to move into concept 2. Concept 54.2 says diversity and trophic structure characterize biological communities. So we're going to widen our scope just a little bit. We have been talking about relationships between two specific species, forms of mutualism, predation, things of that nature. We're going to widen our scope just a bit and start to look at the entire community as a whole and talk about how we can characterize or describe those communities. In general, you're going to find that there tend to be some main species, some primary species, and then some secondary species in any community. There's always a few species in the community that tend to exert the strongest control on that community structure. They tend to be, in a way, the bosses of that community. They tend to have the most influence on that community. Two fundamental features of community structure that we're going to look at are species diversity and feeding relationships. Let's start with species diversity. Of course, species diversity is basically an understanding or an explanation of the variety of organisms that make up a particular community, like the number of different kinds of creatures, the number of unique species within that community. And it has two components or two parts to it. One of those is called species richness and the other is relative abundance. Species richness is the number of different species in the community. How many distinct, separate, individual species are there within that community? And then relative abundance is the numeric proportion of each of those species. For example, you could have one particular community that has 10 different species, but two of those species make up 90% of the population. Well, that might be species rich, but the relative abundance might be a little uneven. Or you could have another population where there's 10 separate species, and each of those 10 species make up about 10% of the entire population of all the living creatures. That would be high in species richness and relative abundance. So you have not only the types of species, but you also have their relative numbers. A good example they use in the textbook is using different varieties of trees or species of trees. You have two forest plots here. You have forest plot on the left, community 1, and then community 2 on the right. And if you were to ask which one of these would you consider more diverse, most people would automatically say community 1 on the left, and that would be correct. Even though there are 1, 2, 3, 4 different species of trees present in each of these communities, you can tell that the the numeric ratio is not very even in community 2. Community 2 is by far of one species. 80% of the trees are of type A in this particular community, where over here you have a very even division amongst all the numbers. So even though the species richness is the same in both of these communities, the relative number or the species abundance is not. And so this would mean that community 1 would be considered more diverse in this case. So two communities can have the same species richness, but different relative abundance. Now, because this is a mathematical equation, we have mathematical formulas that we can use to calculate this. And we have what we call the Shannon Diversity Index. The Shannon Diversity Index uses a number of factors to calculate the diversity of a community based on the number of species and the relative abundance of those species. Now when you see this formula, I don't want you to freak out here because I would not ask you to memorize this formula. I do want you to know what the purpose of the formula is and perhaps kind of a background understanding of how it would work and how it would give us the information. So in here you have various components that we're going to work with. A, B, and C are the species, P is the relative abundance of each species, and N or IN is the natural logarithm. So basically what you're doing is you're just creating a numeric output using those two factors that we just discussed, the relative abundance of each species and then how many species are there. So I'm not going to ask you to know this formula or calculate using this formula. Just understand that the Shannon Diversity Index is a mathematical tool that will allow us to calculate the diversity of a particular community. And what we get is H, which is this index number that gives us the relative diversity of that particular community. If we're talking trees in a forest plot, it's pretty easy to calculate the diversity. We could actually go in and count every single tree and we could come up with numbers that we could put into the index to get us what we want. But a lot of times it's pretty difficult to count the number and abundance of species, especially for tiny organisms or microscopic organisms or inaccessible areas, like trying to count all the microorganisms in a particular pond, all the variety and number. That would obviously be impossible. So what way can scientists use to try to get at these diversity index numbers when you can't just go out and physically count creatures? Well, we use oftentimes some molecular tools to help determine the microbial diversity, or if we're talking about say microbial diversity within soils or within a pond, there's no way we can count those. So we're going to use DNA fragments. So we can take a sample of the soil or a sample of the pond, and basically we're going to sort that by the DNA that we find within that sample. So by examining DNA fragments particular to each species, we can get an estimate of diversity that way. So we're not counting the actual creatures, we're basically looking at the DNA that we get from the sample and then dividing out the types of species that we find based on the unique DNA that we find, and the relative abundance of the species based on the relative abundance of the different kinds of DNA that we find. A good example in your textbook here shows soil pH and the Shannon Diversity Index charted against soil pH, and what you see is that soil pH has a direct impact on the number of microorganisms, the diversity of the microorganisms. Remember, the Shannon Diversity Index not only includes the different number of species, but their relative population counts as well. So you see, if you want to have the greatest variety of microorganisms and the greatest number of microorganisms, your pH of soil should be somewhere in the 6 to 7 range. So right about where neutral would be 7, that's about where things are highest, anywhere from say 6 to 8. You see, the more acidic the soil is, you start to see a big drop off in not only the number of organisms, but the type of microorganisms that can live there. So this is a good indicator that soil pH plays an important role in biodiversity of microorganisms within the soil. When scientists want to study diversity and how diversity affects the stability of a community, if we have the ability to do so, they will oftentimes manipulate that diversity in various experimental plots. They will develop basically separate experimental little communities, and they will actually go in and take out species or add in species to see how that affects the overall community. For example, plant diversity has been manipulated at this place called the Cedar Creek Natural History Area in Minnesota for over 20 years. They have all these separate little plots, and they have the ability to go in and remove species or add species, and they have these long-running, long-term community experiments to see how that diversity affects the community, that community as a whole. So in this case, one of these squares, and this is an aerial photo, so these squares are probably quite a bit larger than you think they are. This is a community. This is a community. This is a community. Every single one of these squares is a separate community, and it's like a little experimental, biome that the scientists can manipulate and study. What we tend to find in these experiments, as well as what we tend to find just by observation out just in the wild natural world, is that communities with higher diversity are almost always better off. They tend to be more productive and more stable. They're better able to withstand the various negative forces of nature, such as disease outbreaks, natural disasters, things of that nature. They're also more resistant to invasive species, creatures that might come from the outside to take over their particular ecosystem or their particular plot of land or area. So when you hear scientists or ecologists, or you hear talk in the news about species diversity and the danger of losing species, you may think, well, what's the big deal if one particular species of frog goes extinct, or some little creature that you never even heard of goes extinct? But as you know inherently, and then certainly as you've studied ecology and biology, you understand that there's food chains and food webs, which we'll talk about more in just a second, where all these species are interconnected. And if you reduce diversity within the ecosystem, it's kind of like playing that little block stacking game Jenga. So as you pull a block, when it's your turn in Jenga, you pull a block and the tower still stands, but it becomes less and less stable over time. So the entire game there is to try not to be the last person who pulls down the last block that causes the tower to fall. So species diversity in an ecosystem, in a community, is pretty much exactly the same. Yes, in the game of Jenga, you can remove a lot of blocks before that tower falls, but with each block removed, the tower becomes less and less stable. Very similar to biological communities, you can remove multiple species sometimes, but with each one, the entire community becomes less stable. You also notice in the game of Jenga, virtually no one pulls from the very bottom of the stack, because those blocks tend to be more integral to the structure of the tower. And there are also species within communities that tend to be more integral to the structure of that community. So there is very much a lot of correlation between those two ideas. The next thing we want to look at is trophic structure. This is something you've studied since elementary school. You've talked about food chains and food webs since elementary school, and that's what trophic structure is all about. It's the feeding relationships between organisms in a community, and it's basically all about the energy flow through a community, the transfer of energy from one creature to the next. Of course, we know that the energy that we're talking about originated from the sun, and then that starts with producers, so that's why plants of some kind are always the beginning of a food chain. They are the only ones who can convert that solar energy into the chemical energy of large molecules that can then be passed on to other creatures. It's a key factor in community dynamics, and of course we're talking about food chains, which are basically links. It's a chart or a picture that links all the trophic levels starting from the producers through the very top apex predators, the carnivores. So all food chains start, of course, with energy from the sun and then various other inputs like water and carbon dioxide from the air. Producers are the ones that capture that energy and turn it into things that other creatures can use, and then we begin to move through the entire food chain. At the end of the food chain, what's interesting is we have to have a way to tie this off or kind of take it back to the beginning, and that's what your decomposers, your fungi, and your various microorganisms, they are the ones that close this loop and get us started again. A couple cool videos there in the notes folder. I think we've seen this one before of the shark eating a seal. Of course, a shark would be considered an apex predator, and he is eating that seal to take in the energy. And of course, that seal, the energy in the seal came from perhaps a fish it ate. The energy in the fish came from, say, the plankton that it ate. And of course, the plankton got its energy from the sun originally. Here's a couple good examples of food chains. No doubt you've seen this type of thing before. A terrestrial or land-based food chain is going to start with some sort of producer, a plant of some kind, and then we're going to move to a primary consumer. Oftentimes a primary consumer is an herbivore, it only eats plants. And then we're going to move up the line to carnivores and omnivores, secondary consumers, tertiary consumers, and quaternary consumers at the very top, the apex predator at the top here. And we also notice another way of looking at this is generally in a pyramid, because it takes multiple plants to support fewer grasshoppers, to support fewer little mice or moles, to support fewer snakes, and then fewer hawks at the top. So you tend to have this pyramid shape, and that also plays a part in the transfer of energy that we're going to talk about, because it's not an efficient transfer of energy. Not all of the energy of the plant gets passed on to the grasshopper and on up the line. In the ocean, here's another good example of a marine food chain. You have the phytoplankton, which acts as the plant, which does photosynthesis. Then perhaps you have some zooplankton, some animal-like microscopic creatures, and then you start with your carnivores up the chain, so producers, and then primary, secondary, tertiary, and quaternary consumers until you reach your apex predator, which in this case is the killer whale. As I'm sure you've studied before, food webs is just another way of looking at that. A food web is basically multiple food chains all integrated together. Probably a bit more realistic way of looking at these trophic structures, because a food web is a branching food chain with complex trophic interactions. So instead of just being a linear single model, you put a bunch of food chains together and form this food web. Like if we go back to this food chain here, it shows the little shrew or the mouse eating the grasshopper. Well, the little shrew or mouse is not the only creature who eats grasshoppers. There are probably 100 other creatures who eat grasshoppers. So this is one example of a linear trophic relationship between these creatures. But in reality, it's quite complex, and you have food webs where there are multiple creatures who feed on multiple others, and you have energy flow between them in a very complex pattern like you see here. What we find when we look at food webs is that it's more complex than just a very simple food chain like an elementary school kid might make, because different species can play a role at more than one trophic level. In some cases you're a predator, in some cases you're prey. It kind of depends on where you are, and you have different parts to play within that food chain. Oftentimes we try to simplify food webs because there's no way we can put potentially all the creatures in a community within a massive food web. It'd be so complex and there'd be so many lines you wouldn't be able to make sense of it. So oftentimes we group species with similar trophic relationships into broad functional groups. So that's where you might get, say, your apex predator. Well, that's kind of a group, and that might be describing multiple species. We also isolate a portion of a community that interacts very little with the rest of the community. So instead of trying to look at the entire complex picture, oftentimes we pick out a portion of that picture. So here's a good example. This is a partial food web for the Chesapeake Bay estuary off the U.S. Atlantic coast. Of course, there are hundreds of other creatures involved in the Chesapeake Bay estuary, but it might be difficult to kind of look at them all together. So here we've kind of picked out a few that have a particular relationship, and we're showing that here in this partial food web. When you look at food chains, you tend to notice, you start to see some patterns, and one of the patterns that you see is most food chains out there in nature are really not that long. From the sun through the plants to the top predator, there tends to be only a certain number of steps. Most food chains in the natural world are five or fewer links long, or 5 or fewer levels, and that's kind of interesting. You might ask, why is that? Anytime you see a pattern in science, which is part of what science is all about, is identifying and finding patterns, you'd want to ask, why do we see this pattern? And there's two hypotheses that attempt to kind of explain this pattern that we see with food chains, and that's the energetic hypothesis and the dynamic stability hypothesis. The first one, the energetic hypothesis, suggests that the length is limited by efficiency and energy transfer. Remember, I said that, say, the grasshopper doesn't get 100% of the energy from the plant. It's actually a very inefficient transfer. It's only about a 10% energy transfer. So a producer level consisting of 100 kilograms of plant material can support about 10 kilograms of herbivore biomass. That would be the total mass of all the individuals in a population. So it might take 100 kilograms of plant to support 10 kilograms of grasshopper. Well, that's only a 10% basically passage of energy, so it's very inefficient, and that's what leads to that pyramid shape that we were talking about. So what that means is, if you put in longer and longer chains, more and more links, you get to the point where you don't have enough energy being passed forward to get to the top of the chain. The dynamic stability hypothesis, on the other hand, proposes that long food chains are inherently less stable than short ones, and that kind of makes sense too, because if you have long food chains, there are multiple opportunities for disruption within there, and then that can kind of disrupt the entire chain. So I think there are portions of both of these hypotheses that help explain food chains and trophic levels, but it seems like most of the evidence tends to kind of support the energetic hypothesis, the fact that the energy transfer is very inefficient and you're going to lead up in this pyramid shape. And that kind of indicates or helps explain why the trophic levels can only be 4 or 5 chain lengths long. A good experiment they talk about in the book is where they're looking at the microorganisms, like in tree knotholes, like when a tree kind of develops a soft spot and there's like an opening within the tree and leaves develop inside there and the rotting material, and there's all little microorganisms that live in this tiny little community. Well, the scientists controlled the amount of leaf litter that were in these tree holes, and that had a direct correlation with the number of trophic levels, the number of links within the food chain. So basically what this is showing us is, when there was plenty of energy from the leaf litter, they added energy to the system could support more links in the chain, and the less energy there was, the fewer links in the chain could there be. So this tends to support this idea that it's all about the energy and the energy transfer, and that's what limits the length of the food chain. We're going to take a quick break right here. When we come back, we're going to talk about the fact that not all species within the community have the same importance necessarily. Some of them tend to have a larger impact than others. So we'll take a break and we'll come right back for that.